Okay. So this is a picture that was made by an adolescent boy with autism, and he called it the Tree of Life. And so if you look really closely, you'll see that the fruit of this tree are little, these are little wombs with a little fetus and an umbilical cord reaching up into the stem, the branch, and coming from the tree. And that at the root, the roots of this tree is the kind of the trash of our modern society. And I really fell in love with this picture and felt like it really summarizes kind of my research focus. And so I talked to I actually never met him, but the psychologist who worked with him negotiated that I could buy this. He may have actually given this to me. The picture, not the picture itself, but at least I was able to use it in my presentations. So, oh wait, I can use this. So environmental epidemiology of autism is the title of the talk, and I'll be going through some of the literature a little bit and our own work from the CHARGE study at UC Davis. So just as background, what is autism? It's a developmental disability. It was originally described in 1943 by Lee O'Connor from Johns Hopkins. And the way he described it is actually very similar to the way we diagnose it today, which is there are deficits in several different domains. The earlier ICDs used just the first three of these that are here: the social reciprocity, the communication, and the repetitive behaviors or restricted interests. The newest definition now includes sensory sensitivities in the US DSM, which is the Diagnostic and Statistical Manual, and I think that that's also happened with the change in ICD, but I'm actually not certain of that. The symptoms are generally present by 36 months of age, and it's been considered to be a lifelong condition, although there is debate about that. There are some seats up here if people want to sit down who are standing in the back. It's going to be a long stand. It's diagnosed primarily through two instruments, but particularly the top one here, the Autism Diagnostic Observation Scale, which involves a semi-structured play interaction. And I actually have a video clip I'll show a little bit later of one of those tests being done. And then also interviews with the primary caregiver, which cover the history of the child's developmental trajectory and milestones. And both of these provide separate scores for the different domains. These are some basic facts. It's a very male-dominated field, or actually diagnosis, and about 4-to-1 males to females. In the U.S., the current prevalence seems to be about 1 in 59. And historically, there was a long period of time after the condition was described when it was considered to be due to bad parenting. There was a phrase called refrigerator moms. And I think there's still a legacy of this kind of blame the victim thinking, even though nobody believes that at this point in time. And we certainly understand that there is this neurological basis in aberrant brain development. And that really derives from post-mortem autopsy studies as well as electrophysiology, and then more recently MRI studies. So it's highly heritable. Twin studies, family recurrence, and candidate gene studies have all shown that. And of course, there's a lot of, at this point in time, a lot of genome-wide studies confirming that. So environment and neurodevelopment, it's not a new topic. In fact, in the 19th century, in the late 1800s, actually in the early 1800s, it was already known that lead was harmful to children. Some of the English language literature in the early 1900s documents the convulsions, the abdominal distress, and this was from high-level exposures. And there were recommendations for removing lead from paint, particularly in cribs and toys, children's toys. The turning point in terms of low-level lead exposures was actually in 1979 with this landmark paper that used deciduous teeth and measured the lead levels and showed that early lead exposures were strongly correlated with child's IQ, language processing, behavioral problems, and reaction times. And that really changed the way people thought about lead, although Herbert Needleman was assailed in a really despicable way by the chemical industry. And today it's really a political issue more than a scientific issue in terms of any controversy. And unfortunately, we still have a lot of communities that do have a lot of lead from legacy paint and water issues. Similar kind of story for mercury, where high-level exposures were documented to have really horrendous effects. This was the dumping of chemicals from a plant into the Minamata Bay in Japan. Thousands of people died from this, and they spent decades trying to figure out what the cause was, and eventually, in the late 60s, figured out that it had to do with the mercury in the bay. Many children were born with mental disabilities as well as physical malformations. And now we know that lower levels also bring on cognitive impairments. So another disaster, what seemed to be something of a disaster, was going on in the 90s related to autism, and the rates of autism were climbing really rapidly towards the end of the 1990s, beginning of the 2000s. We looked at those data and did an age cohort analysis. So along the x-axis you have the age, and each line represents a different birth cohort. The fact that these lines do not crisscross is evidence that there's a trend here, and you can see each successive birth cohort has higher levels, higher incidence of autism at every age. And in fact, it's about a 7-fold difference between the 2001 cohort and the 1990 birth cohort. That's a 600% rise, and when we looked at possible explanations, we found that artifacts related to diagnosis, better diagnosis, more awareness, earlier age at diagnosis, changing criteria, could only explain about 1/3 of that rise, leaving open the question of what's, to the extent that there is, part of this is a real rise, what are the causes? And so let me take a step back and just say what I mean by causes. I'm looking at root causes, incipient causes, what actually initiates that pathogenic process. Now, there are factors that may contribute to the severity. It may contribute even to the duration of time for onset. And some of those exacerbating or mitigating factors might include the same factors that are initiating factors or not. But all of my work is really focusing on these root causes. Sometimes when I talk to families, they're thinking about what makes their kid do better one day to the next. So that distinction is important. And the other aspect of causation is that it is multifactorial, and particularly when you're talking about really complex disorders like autism or lots of physical health problems as well, there's probably multiple causes. Very few things are really unicausal. So these pies represent, each pie of different factors here represents a set of sufficient causes, and that applies to the individual as well as across the population. So there may be multiple different sets of sufficient causes that can actually bring about the full manifestation of symptoms that leads to a diagnosis. And this is kind of a really rough timeline, and I think really it's not necessarily to read this slide, but just to say that in the beginning there really wasn't a whole lot for decades. And that earlier work by Bruno Bettelheim that claimed that it was bad parenting came out in the 1960s, and it really dominated the field for many decades, even though there were other people pointing to environmental aspects, including kind of the family environment, but also there was early evidence of congenital rubella having a very, very strong effect. There was an epidemic of rubella in the 1960s, and the rates of autism in that population of people who were seen clinically for rubella was somewhere close to 10% of the children who were showing these autistic symptoms. Thalidomide was also linked to autism. And so these tan ones are the kind of medical related exposures. Then we start seeing actual, what we call environmental, from the point of view of chemical exposures, exogenous kinds of factors that are not infectious, playing a role. The literature sort of cropping up in the mid 2000, about 10 to 12 years ago. And this is really most of what there is about etiology in the early period. These are the genetic studies, and of course the genetic studies could fill this room with starting in about early 2000s. But then the environmental field began to take off. And over here, these are only a smattering of the studies, whereas on the left, that's really all there was. I've already made this point, and there are various ways in which genes seem to contribute to autism, and then we have what we breathe, what we eat, what we drink, medications, household products, and personal care products. The point here being that these environmental factors actually provide us with a way to intervene. So that's where the public health impetus comes. Genetics is not really leading us in that direction. Now the bad news is that there are tons of different chemicals out there. In the United States, over 80,000 different chemicals have been licensed for commercial use. Thousands of them certainly are in use, maybe not all of them. And the vast majority have never been tested for neurodevelopmental impacts. The vast majority have never been tested with early life exposures, including beginning during the prenatal period. So that's a problem. And in fact, when measurements are done of either maternal blood during pregnancy or cord blood, a large number of chemicals are detected of the ones that are looked at. So one study found about a third of the tested chemicals were present at birth in those children or in the pregnant women. So the good news, though, is that multifactorial causation actually gives us a little bit of hope about reducing the incidence of these conditions. And the reason is because the concept of a sufficient cause means that if you take out one of those factors that contributes to what is in total is a sufficient cause, this is now insufficient. Now that doesn't mean there might not be some impairments in that child, but they're not at the level of a clinical diagnosis. So in reality, if there are certain factors that tend to appear in many of those pies of sufficient causes, hitting at those factors alone may provide the opportunity to really reduce the risks overall in the population. And that's important because, I talk to my colleagues who are in neuropsych and, work on child development, and their eyes start to cloud over when I talk about environment. They just say, this is too terrible. I can't be worried all the time about what I'm eating and what I'm breathing and where I'm sitting. And and I agree, it's a big burden. The population, people should not have to worry. People should not have to do what I do all the time, which is read every single label of everything I eat and buy and so forth. That's why we need public health policy. That's why we need governmental action, societal commitment to reducing these chemicals. Okay, so moving to the research that I've been doing. So this is a study I launched when I came to UC Davis in 2002 to look at autism and environmental contributions. And at that point, really, the only things that were known were the ones that I showed in that big timeline slide, which were thalidomide, also valproic acid, congenital rubella, and some candidate gene studies. And basically, somebody asked me that, if I would join this center that was focused on environment and neurodevelopment, specifically autism, they were writing the grant. And I said, well, I don't know anything about autism. And they said, oh, that's fine, nobody does. So, they sent me a book on the neurobiology. I read a couple of chapters. I thought, oh, this is really, pretty interesting. And then I basically reviewed the literature in 1 week, and it was, really there wasn't much there. So that's why I took a look, and from basic epidemiology, you learn that if you're kind of in the beginning of a field and there's not much known, a case control study is a good way to start, right? You can cast a wide net. So our goal really was it seemed modest, to identify causes and contributing factors for autism and also for other developmental delays. And we were interested in environment, also genetics, but not singly genetics as it interacted with environmental factors. So the study design was to have 3 groups: the autism group, the developmental delay group, and a population-based control group of children with, primarily typical development. And here we matched on age, sex, and broad geography. The way we got those, the children with the developmental conditions, was through the California State has a Department of Developmental Services, and they had a database of all the people who received services through state funding. So we've collaborated with them, and they have provided us with lists of people that we can then contact. So I feel very lucky because I don't know very many places that have this. And it's not 100% population coverage, but it's pretty high. It's probably in the 80% or higher. And when we started, there was actually a paper showing that about 75% of children who were diagnosed by school age or at school age were covered in that system. And then we use the birth files to get the population controls. I hadn't thought of it this way, but my colleagues at the Mind Institute pointed out that the developmental delay group could also be looked at as a control group for the ASD. And in fact, that allows us to say, is this finding specific for autism, or is it a general risk factor for developmental delay? And so we do have data that allows us to do that. These are the eligibility criteria. We wanted to get the families when the child was fairly young so that the mom could remember her pregnancy pretty well. And we took English and Spanish-speaking families. So we do the assessment with the 2 instruments I mentioned earlier, and we also do a medical exam and some other developmental tests, the Mullen Scale of Early Learning and the Aberrant Behavior Checklist and the Vineland Adaptive Behavior Scales on all of the children, actually. The only thing that we do just on the ASD children is the ADOS and the ADIR. We also screen those other 2 groups to see if they do have symptoms that they may actually have autism that had gone undiagnosed. So we use a screening, the social communications questionnaire for that group. So this is actually an administration of the ADOS. The tester has just set up that there's a birthday for this little doll here, and has a little Play-Doh cake, and you'll hear what goes on.
Ready? Let's light them. Oh, it's hot. Oh, hot. Ready? Let's say, happy birthday to you. Happy birthday to you.
You can see he's kind of ignoring her and just interested a little bit in the piece of Play-Doh. So this is pretend play that he's not engaging.
Okay, time for cake. Baby's so hungry.
And by this age, 2.5 or so, children have been to birthday parties, they've sung the song, they kind of know the routine.
Can you give baby some cake? He's hungry. Here you go, baby. Here you go.
Oh, whoops, sorry. Oh, I didn't mean to do that. Let's see if it picks up.
Light the candles. Ready?
No, okay, sorry. So what actually happens is he drops the piece of Play-Doh, he goes down, he picks it up, he puts it on the spoon. She's just said, let's feed the baby. He takes it and he starts to put it in his mouth. So he's not even getting that this isn't real cake. It's just a play, representation of the cake in the party. So this is a whole level of not understanding the social cues, not understanding what's really happening and not being able to abstract from reality and play. So the study is being done at the Mind Institute, which is located in Sacramento in California. And when we started the study, we started with this list of potential neurotoxins that might be related to autism. So pesticides we know affect the nervous system. Metals due to some organic pollutants had been associated with cognitive delays. And then there was literature about infections, and there were hypotheses about increasing use of psychotropic medications and then nutritional factors. So those were sort of our major things that we were interested in looking at. And on the right side are these different ways of collecting information to measure those exposures. So these pesticides, we actually have a database in California. We have a lot of nice databases, but this one is all commercial pesticide applications are recorded and reported to the state. We also do a questionnaire where we collect from the interview their use of consumer products, including pesticides. And then we have the residential history, which is what we link to those databases. Metals can be measured in lots of different material. And baby locks, we asked the mothers to bring in the first haircut. And we're now collecting deciduous teeth. So these are some of the different ways that we collect different types of information. Of course, it's retrospective, and that's the disadvantage of a case control study. But when you're looking at existing databases that actually report, say the pesticides, that's not really of concern, and we know people do know where they've lived. They can give pretty good information about their residential history. So that's the approach. And then we're interested primarily in the early life, the prenatal period. We actually start with 3 months before conception through the pregnancy, and we do collect later data, although most of our analyses have focused on the prenatal and sometimes the first year of life. Okay, turning to some study results. I'll start a little bit with pesticides, and those of you who are coming to the journal club tomorrow, we'll talk more about this. So insecticides in all species, most insecticides in most species act on the nervous system, and they actually interfere with ion channels. It can be calcium channels, sodium channels, and so forth, involved in neurotransmission. They may have other impacts as well. And many of these pesticides were actually originally developed as nerve gases, the organophosphates, for example, for war around World War II. Were exposed through food residues, household applications, the pet, flea, and tick treatments, and then broad spraying in the home, and then secondary exposures through living near agriculture or from public health applications. We get sprayed a lot in California because of various problems with insect pests that affect crops. So they do a lot of spraying. So this is a summary of papers looking at organophosphate pesticide exposures and looking at things like IQ as well as some behavioral outcomes. And these are just a smattering because there are actually there's probably about 36 studies now on OP pesticides and neurodevelopment, and almost all of them, 90% of them, are showing associations with deficits. There's also some data that came out of Columbia University looking at volumetric measures in the brain and showing that there are some associations between chlorpyrifos exposure, one of the organophosphate pesticides that is currently supposed to be getting banned in the U.S. and it's been met with a little bit of resistance. But this was associated with changes in areas of the brain that are involved in attention, in language processing, in inhibitory control, and social cognition. So areas that are relevant for autism and of course ADHD and other developmental disorders. So we linked people's addresses. In the middle of this circle is like a home. So we draw these buffers around the home, and then we link that to the pesticide use data, which is recorded in these units, which are the squares on this map. And we looked at several different classes of pesticides. This is from multiple logistic regression models, adjusting for all the things on the right. And what you see in this figure is that the organophosphates are in the top panel. These are 3 different buffer zones of 1.25 kilometers, 1.5, and 1.75 kilometers in each box. And what you see is that there's some elevated risks from second trimester exposures in particular, but also for the whole pregnancy for organophosphates. And then the strongest association actually is in the, is in chlorpyrifos in the second trimester. Actually, the third trimester for overall organophosphates is stronger. And then the pyrethroids, and I just want to say that for the chlorpyrifos, it's actually, those are odds ratios of between 3- and 4-fold. And then the pyrethroids, there's a preconception association with autism. There's a little bit of pushback. Some people wrote this letter to the editor pointing out that we didn't have confirmation of exposure or direct evidence that the active ingredients drifted onto the resonances, or that they were inhaled or ingested. And they claim that they've already looked at this, and that ground spray drift only deposits 1/100,000th of the applied amounts at 0.4 kilometers downwind. However, there was a study that came out that same year that was done in California, looking at that database that we use to assign exposures, and so that's what the bars are. The weekly average. Oh no, sorry. It's the solid circles are the applied amount per week in kilograms, and this was for chlorpyrifos, I think, actually, and so this is amount applied per week, and this is a whole year, 52 weeks, and then the bars are what was detected in air samples that were put on top of schools that were nearby. And you see that there's a pretty good correlation here between when there are applications and how much those applications are and what's actually being measured in the air. I would argue that if it's in the air, people are probably breathing it. And that this is actually a pretty good validation of that database and what's in there and how we were using it. There's been some other studies actually also looking either at ASD diagnosis or symptoms of autism. This is PDD, stands for pervasive developmental delay. These are, so there's a few studies out there now looking at organophosphates in relation to either an autism diagnosis or symptoms. So one of the, just sort of ending the pesticide part of this talk, these are, this is just to say that we have this, this vicious cycle where we introduce a group of chemicals. Over time, evidence starts to accumulate that they're harmful. Meanwhile, the chemical industry is busily working on synthesizing new chemicals that they can replace those with. The new ones get put in, and over time we start to realize what's wrong with that they are also harmful to health. So this is really a problem, and it's not just true of pesticides. We're seeing it with plasticizers. There's no more BPA in our plastic water bottles in the US, but now it's BPS. So how much better is that? And, you know, all of the fuss over just chlorpyrifos when there are all these other organophosphate pesticides, I'm a little, you know, we have to fight over chlorpyrifos, but getting that banned, but that's only the beginning. And now these pyrethroids, and we have some evidence that there may be issues of neurodevelopmental disorders associated with those. The neonicotinoids, of course, are the ones that are really wreaking havoc on the bee colony populations. So there has to be another way. Instead of replacing one toxin with another toxin, we need to be looking at other sustainable ways of controlling pests in agriculture and doing agriculture. It doesn't need to be large monocrops everywhere. So the next set of results are on air pollution. And we certainly know that air pollution does lots of awful things to the respiratory system and the cardiovascular system as well. And there's some evidence that neurologic and neurodevelopmental disorders may also be impacted. So we took some of the residential data and we derived two metrics. One is a measure of just their distance of the household to the nearest freeway, to look at as a marker for traffic-related air pollution. And then we also mapped to these models that have been done. Some of these models have been validated in large data sets across the United States, and they give us estimates of coarse and fine particles, NO2, ozone, and some other pollutants. And so that's how we did this. So this table shows that if we compare to people who live the furthest distance, the bottom of the exposure level from using distance, so that's our reference group. It's about 9/10 of a mile, a bit more than a kilometer and a third or so, and we compare to some of these other groups that are closer. We find that the closest group, the closest 10%, which were 2/10 of a mile or 300 meters, had an elevated odds of being cases as compared to controls. There was no evidence of an increase in the middle groups, and I'll come back to that. And then looking at the actual estimated modeled exposures, we see that there was an elevated odds ratio for the first year of life and for the pregnancy period of around twofold, even after adjustment for sociodemographic factors, and somewhat lower risks during each of the trimesters separately. So we were seeing an association both with this distance to freeway and the regional air pollutant measurements, with cases having higher than the controls. As far as biological plausibility goes, we have an objective measure of exposure, and the particle concentrations are actually known to be highest when you're really close to the freeways. It drops off to background levels at about 300 meters, which was the cut point for our top group. So the lack of a dose response is exactly what we would have expected, just knowing what the actual exposures are at those distances. There's a pretty good-sized toxicologic literature of studies of neurodevelopmental deficits in rodents, and a lot of work being done on mechanisms by which air pollutants can actually get into the brain, involving the olfactory nerve and all sorts of other interesting mechanisms. There's also PAHs are known to induce oxidative stress. They can be endocrine disrupting and induce inflammation. And inflammation actually is one of the theories about autism, is that it's a neuroinflammatory process going on in the brain. And a lot of immunologic studies have shown that people with autism have immune systems that look a little bit different in terms of the cytokines and chemokines that they produce and how they respond. Sometimes they're at a sort of more revved up state of immunologic surveillance, and then they don't respond as well when they're actually challenged. So that's actually part of the mechanistic research that's been going on. And then there's also epigenetics. So the fine particles have been associated with hypomethylation of DNA in the placenta, a recent finding. And there's now a huge number of studies looking at air pollution and autism. Many of them do show associations. I was putting in italics the ones that don't. So there are these two from Europe, and also the Kalkbrenner is a negative study as well. But most of the studies are showing these associations. Have we established it's causal? I'm skeptical still. And partly because these are really good studies that were done in Europe, and my impression is that the socio-demographic gradient in terms of air pollutant exposures, they're very highly correlated in the United States. I don't know what the situation is in Canada, but my impression in Europe is that there's a lot of wealthy people who are living in centers of cities and maybe getting some of the higher exposures. So there could be confounding going on, even though many of these studies did a pretty good job of trying to control for that socio-demographic factors as confounders. However, I think there's another way to look at this, which is that it may well be that we're talking about susceptible populations to the air pollution, and that these socio-demographic factors are actually potentiating the impact of the air pollution, and that we really, again, going back to those causal pies, we really need to look at the complex of exposures. And maybe it is that wealthier people who have better access to health care and better nutrition and fewer liquor stores in their community and so forth are the ones that had the resilience and aren't affected by the air pollution. So sometimes we always think of confounding, is it confounding, is it confounding? But there could be heterogeneity, which is actually at the root of the differences across these studies. So the third one I'll talk about is folic acid. So folic acid is really required for repair of DNA and synthesis and repair of DNA. We recommend supplementation because supplementation actually prevents neural tube defects, and more and more women do take these supplements, but they need to be taking them very early in pregnancy. And actually, since supplementation and fortification of certain cereals and grains in the U.S. and in some countries don't fortify. I don't know what Canada does, but there has been a reduction in neural tube defects. So we looked at folic acid as a potential protective factor in autism and calculated estimates of the intake from the different things that are shown here, the multivitamins and cereal and so forth, and then did a logistic regression analysis again and we screened for a lot of confounders. We actually only found two that confounded, so these are not as, they're sort of very parsimonious models with maternal education and birth year. And this is looking at each month, the 3 months before pregnancy and then the months of pregnancy, and what people self-reported in terms of use or not of prenatal vitamin supplements, of which folic acid are a major ingredient. We found that for the 3 months before and the first month of pregnancy, there were a higher proportion of the control mothers who were taking those prenatal vitamins as compared to the case mothers, with an odds ratio of 0.6. So a 40% reduction in risk. Pretty strong effect. So this was published in 2011, and of course one of the issues was whether this was a self-report recall bias type of thing. So this would have had to have been recall bias in which cases were less likely to report taking their folic acid than controls. Usually in recall bias, studies have looked at recall bias, people report more of things, all kinds of things. So it sort of doesn't quite fit that story. But we also saw a dose response. So the higher the amount of folic acid, the more likely they were to take the prenatal vitamin supplements, the less likely they were to develop autism. And there was, at the time that we were publishing this, and there were still some people who argued that we are actually over-methylating people because folic acid is a methyl donor, and that itself was causing autism. So we went in with sort of the two-sided hypothesis of was there a problem with too much or too little, and we didn't see evidence of that. And there's still some people who are arguing that high levels are harmful, and I've heard them present at meetings. So the really exciting thing for us was when our findings were replicated a few years later in this Norwegian large, population-based cohort with thousands, tens of thousands of children, where the reporting was done during the pregnancy, so the mother obviously did not know the outcome for her child. And they came out with an odds ratio that was almost spot on the same as ours of 0.6. So this was kind of validating, it felt like. And there have been some other studies. There is a study that did not replicate this. I have things I could say about that study, but I do think that the evidence is really starting to accumulate. And later, a little bit later, a few more slides down, I'll talk a little bit about the gene-environment interaction work that we did on this study. So the very last kind of main effect issue that we looked at was maternal diabetes and metabolic conditions, the three here: obesity, diabetes, and underlying hypertension. And these results showed that if they had any one of these, there was a 1.6-fold higher risk for autism spectrum disorder versus the typicals. And also we found results in the developmental delay group as well. I haven't been showing the developmental delay results, but actually most of the things that we see for ASD we also saw for developmental delay. And here we also saw effects for obesity. The diabetes did not reach significance, but it is elevated, and hypertension. We actually had very few who had chronic hypertension, although we then went on and found that preeclampsia was strongly associated with autism. So before we did that study, there had been a meta-analysis of some mostly small studies that had shown a relative risk in the cohort studies of 1.5 and 1.7 in case control studies, but many of those studies were not very large. After we published, there have now been multiple very large studies confirming these associations. And I'll just point out that when there's gestational diabetes with obesity, and in several of these studies, you see these very large odds ratios: 2.5, 3.9, 3.0, 4.4. Yeah, I think this one actually, I'm not sure whether this one included obesity or not. But they tend to be higher. When there's not obesity being taken co-occurring, the odds ratios are more in this 1.6, 1.4 range, which is actually right where ours was at 1.5. So it looks like diabetes without obesity has some elevated risk, with obesity an even stronger, substantially stronger impact. And there's actually now a new study out that Zhang, the top one, the Kaiser, the one with 300,000 people in it, she's now looked at in that Kaiser database, where of course they've got medical diagnoses of both the exposure and the outcome. So these are really well done studies. She did a kind of a dose response of diabetes, where she took those with underlying diabetes by itself, gestational diabetes, an abnormal level in the glucose tolerance test, and then the referent group of none of the above, and progressively higher odds ratios as you go up that more serious type of condition. So really beautiful work. So just a kind of little thought experiment is, let's look at all of these things that are kind of rising with these time trends. And what brings to my mind is this question that, asthma, obesity, diabetes, certain cancers, asthma, mental health disorders, is there some sort of a common factor that provides, a sort of unifying theory about changes that have been happening in our societies that are resulting in these very specific kinds of syndromes that are all increasing pretty much in parallel. And I mean, the diabetes is kind of an interesting one because you've got the diabetes and then you have maybe the consequences of the diabetes on the offspring, or you know, that metabolic, the metabolic disorders that go with diabetes and obesity, which might actually have something to do with all of this. And we can also think about that inflammation issue and to what extent is there inflammation going on in multiple outcomes that sort of may provide some kind of a mechanistic, continuity across these. So I have two slides that are kind of summarizing evidence about a lot of factors across the literature. And this is totally subjective. Those little plus signs are my interpretation of the literature, and I don't claim to necessarily be correct on these, but this is just a gestalt of how we might think about where the science is today. And in some of these I debate over, I come back to the slide a year or two later and I think, maybe that one goes up or that one goes down. But I put pesticides and air pollution at kind of 2 pluses, that there's some evidence there we really need to be paying attention. Is it totally proven? We need more studies, just because 2 or 3 studies is it's good, but and maybe it's enough even to act when we can prevent things, when it's a public health issue, but it's not overwhelming. So that's where air pollution, pesticides is. And one of the big things that worries me about air pollution is that the smoking literature has been really null. I mean, there's some studies showing protection, there's studies showing effects, and there's been 2 meta-analyses. They're probably all meta-analyses of pretty much the same studies, 15 studies that come out with a meta-analytic odds ratio of 1.02. So that bothers me because the constituents of cigarette smoke and the constituents of air pollution overlap in a big way. And it makes me a little, confused. So there's different things here. Maternal and paternal age is a very large literature, and they both are associated with higher risk for autism, possibly for different mechanistic reasons. And I view these as proxies because I don't think it's the age of the parent per se, but it could be cumulative exposures. It could be other things that change with age, metabolically. And, for maternal age, we know that the eggs themselves, the ova, are more likely to have all kinds of defects in later maternal age. And paternal age, there seem to be epigenetic things going on. Season of birth or conception, is associated with lots of things that are seasonal. Infectious diseases, different nutrition because of what's available to eat. These are the protective ones, the ones in blue, the folic acid, iron, and breastfeeding. And then over here we've got a few others. These are some of the older ones that had odds ratios of 50 or so. Selective serotonin reuptake inhibitors, there's actually a number of studies, but one of the issues is if people are taking SSRIs, they probably have these mood disorders that may themselves be actually the factor that could be detrimental to neurodevelopment of the child. And then congenital rubella is also in that category of old, large studies or not necessarily large, but strong effects. Fever and inflammation has been associated. This is one where I'm almost ready to put a 3rd plus on that one, and I recently added another plus to diabetes here. Preeclampsia and obesity by itself, not sure. And then I have a few slides here about interaction, and I'm going to go through them quickly because I want to leave a little time for questions. But heritability estimates for autism have gone down, although when you go to meetings, people still are quoting this 90% number an awful lot. But these are the larger studies that put heritability at 38 to 60%. And in fact, when people are doing these estimates of heritability, they may be including, did the gene-environment interactions in their estimate of the genetics, because that's how they do it. They determine, you know, what is the concordance in monozygotic twins compared to dizygotic twins, compared to, you know, people who are further apart in relationships, and it doesn't leave room really for the gene-environment interaction, or it makes a lot of assumptions, the models that they fit. And there are actually several, there have been a few studies, but the ones of autism risk and looking at the synergy of combined exposures, these have all come from our group, actually, from the CHARGE study. And so there's these two studies that were done looking at the prenatal vitamins and the folic acid intake, showing that those who have inefficient conversion of folic acid to folate, the active usable form, are the ones for whom the prenatal vitamins are truly protective. It actually doesn't matter for people who have a efficient metabolism due to their genes, including MTHFR. And then this is a gene that's involved in that actually seems to interact with air pollution. And then this study was looking at total burden of copy number variants. So there are segments of the DNA that get repeated because when DNA is replicating, it makes a lot of errors along the way, and sometimes those just get repaired really well, and sometimes they don't. And so you've got individuals who may have whole segments of DNA that might be repeated 50 times, 100 times, and the genes in that region then may not actually express properly. So this was looking at kind of summing up the lengths of all of the copy number variants in the genome. And then these are two studies just looking at the severity of autism, because they didn't have any controls, and they found ultrasound and maternal infection interacting with copy number. Anyway, so this was the MTHFR finding. If the mom had one of the T alleles, meaning inefficient conversion to folate, the risk was reduced by 50%. The same if the child did. If they both did, the risk was reduced about 70%. So odds ratio of 0.3. And so there's different ways to think about gene-environment interaction. There's the combined effects having a synergistic impact. And then there's also environment influencing gene expression through epigenetics, perhaps, and also metabolomics can be changed. That might be the way in which there's an interaction. And you can have both of these happening at once. You could have changes in gene expression from the exposure, but also a synergistic effect in combination with genes. And actually, this is just sort of a possibility also is that, and we did see, there's some data, we haven't quite gotten to the point of publication yet, but we are seeing what looks like an impact, or at least an association of exposure with gene expression. But one of the possibilities is that those, and that was in ASD children, is that the brains in children who are susceptible to developing ASD might actually be influencing the way in which the exposure result affects gene expression. So it could be a kind of reverse causation going on as well. We need to be getting data longitudinally to study that. And we have other studies now, which I'm not going to talk about because I'm running out of time, but looking at a prospective cohort of younger siblings of children with autism who are at extremely high risk for developing autism. So lots of people involved in this. This is our staff, and the families, over 2,000 families have participated, and these are our funders, and thank you.